Bonjour, Professor Malone. We have the pleasure of having Professor Malone, who is the inventor of the RNA messenger technology. And we thought it was interesting, given the fact that we had the opportunity to talk to many doctors, many geneticians, virologists, and so on and so forth. Everybody is talking about RNA technology. We thought it would be much better to ask, I would say, to get the information from the horse's mouth. And I understand that you live in Virginia and therefore you're breeding horses. So obviously the metaphor is of importance to you. But maybe, Dr. Malone, if you could say a few words about yourself, how did you come about this discovery before we delve into the very important matters of, I would say, the RNA messenger vaccine, the issues and some of the messages that you have voiced out either on media or on the internet.
Thank you very much. So a little bit about myself. I'm a physician scientist. People comment, how come you suddenly popped up? I'm a consultant. I do a lot of consulting for industry and for the US government, particularly the Department of Defense. My clients don't typically want me to have a high profile media presence, and I don't seek it. I haven't sought it for many years. Sometimes I speak to the media in background, trying to help folks like you make sure that you do the best that I can to make sure that you get the science right and that you understand the issues. In the current situation, a cluster of events have kind of outed me, forced me onto the world stage, and I'm suddenly inundated, but glad to speak to the press because I believe that the issues are not trivial. They're complicated, and it seems that there's a great hunger, both for people that are willing to speak out and openly. There's much censorship going on, and I have colleagues in France that have written to me many times over the last few months with stories that I personally find alarming, and colleagues all over the world that are sending me information and sharing their papers and opinions, hoping that I might be able to give voice to some of the things that they're concerned about. So I kind of feel like I have a bit of a moral responsibility. In terms of the background, how did this all start, the mRNA story? 30 years ago, I was a graduate student. I had come out of a undergraduate lab that had been very involved in the initial discoveries of the role of retroviruses in non-human primates causing immunodeficiency syndrome. It was a lab that was visited fairly frequently by a eminent French scientist, Dr. Luc Montagnier. Murray Gardner was my mentor. So I kind of started in a world of retroviruses working on mouse memory tumor virus and familiar with the whole LAV, which then became HIV story. That was kind of my origin, was that environment. I was very passionate about retroviruses and sought out an opportunity to apply what I thought when I was a MD PhD student, what I thought was the most obvious application for retroviral biology and understanding of that, which at the time was gene therapy using retroviral vectors. I was accepted into UC San Diego as a graduate student and I had applied there because there were two of the leading gene therapy researchers in the world, Ted Friedman, who had come up with the idea, and Inder Verma, who was at the Salk Institute. Inder was running a highly competitive postdoctoral lab, and there's many things about Inder that are in the literature and science, and I don't think we need to go there. But Inder was trained by David Baltimore, so very knowledgeable retrovirologist, Inder is. And I was fortunate to be the only graduate student in a very large competitive postdoctoral lab. The story, kind of the origin story of this whole thing traces back to about 1988. And oddly, it's not just the RNA story, it's also the use of adenoviral vectors for vaccines. Comes from the same laboratory where Dinko Valerio, later founder of Crucell, was the senior postdoc. What happened there at the time, we were all true believers in gene therapy, very passionate, so was I. The postdoc that I was working most closely with, Dan St. Louis, was working on a technology involving transducing with a retroviral vector murine cells, mouse cells, and then reimplanting them. I'm skipping the details back into mice and looking for long-term expression of a transgene. Simple stuff, straightforward. We all understand that idea. He ran into a very odd problem that no one in the laboratory could understand. After about 3 weeks, the gene expression was lost. You could no longer detect the secreted transgene protein. And since it's also Inder's lab was a leading transcription control lab, everybody went down the rabbit hole of sophisticated theories about gene silencing and retroviral vectors and all that. Being a half-trained medical student who had come out of a vaccinology environment, I said, 3 weeks, that pretty much mirrors the expected time course for generating an immune response against the transgene. Lo and behold, that turns out to be the case, and that paper was published with Inder and Dan St. Louis, and I was mentioned in the acknowledgements, but that was the genesis of this. And appreciate, it was heresy to advance the idea that there was a problem with gene therapy, which was immune response against the transgene, because it called into question the whole logic. But we now know with the benefit of time that that is the case, and it is one of the fundamental flaws of gene therapy. Now, that was followed by, in my own research, that had focused on asking questions about the structure of retroviral RNAs and the packaging sequence. I sought to develop a technology to transfer RNAs into packaging cell lines for retroviruses, so I could ask structure function questions about those RNAs. And I was doing molecular modeling on a supercomputer and other things about RNA. And so this required me to develop a technology that would allow me to efficiently deliver RNA into cell, and the whole suite of technologies, which didn't exist at the time really, for large scale preparation, synthesis, purification, etc. That pretty well mirrors what's being done now, only at a much larger scale. Cap incorporation, the importance of having a 5' and 3' untranslated region, a polyadenylated sequence, etc. And I, as the good graduate student, ran into this system whereby I had developed the RNA manufacturer. I acquired through a collaboration a Xenopus laevis, so African clawed frog transcription vector that would make chloramphenicol acetyltransferase, swap that out with the luciferase RNA encoding protein and sequence, and started asking questions with what was then a very sensitive system about, could you use the standard methods for transfecting RNA that you use for DNA, etc. Nothing was working very well. Tony Hunter, a notable scientist at the Salk, suggested to me that I try a new technology that had been developed at Syntex. And the paper actually wasn't published yet. Phil Fogner's on it and Gordon Ringgold. Tony recommended I talk to Gordon, and that was the lipofectin paper. Turns out that Syntex had had problems getting lipofectin to work with RNA. They'd thought about it, they'd tried it, but they hadn't spent all the time to build the system and the reagents and the methods and stuff that I had done. So I got shipped the lipofectin from Syntex, and it worked on the very first experiment, and then I was off to the races. Suddenly I had a new method for delivering RNA using cationic lipids, and I'd built all the infrastructure to be able to characterize it and express it and monitor it, etc. Tried that on a large number of cell lines, and then another one of these happenstance things that happens in science. I was teaching an embryology course as a graduate student teaching assistant, and I had a lot of extra, we call them tadpoles, frog embryos. And it was a very simple, I wonder what happens if I add this to that. And then shortly thereafter, suddenly I was transfecting whole animals and getting gene expression from the RNA, protein expression from the RNA in Xenopus laevis tadpoles. I then went on and tested it using DNA in chick embryos, and that also worked. So at that point, I had an in vivo delivery system and a lot of background with different cell lines in situ. And the whole build of what's necessary for RNA structure to make this work, how to manufacture it and purify it at milligram quantities, and this had never really been done before. So then the patents started getting filed, and the argument started over who owned the patents, and was it the university or was it the Salk, and could I talk to this person or that person? And it became so untenable, I had to leave the Salk. Frankly, I left with a diagnosis of post-traumatic stress disorder. It was extremely unpleasant. We don't need to go there right now. The person that I'd been collaborating with had just been hired from Syntex because he was frustrated. Syntex wouldn't take his discovery of cationic lipids forward. And he joined a little startup, Bill Feldner is his name, called Vical, which was almost across the street from the Salk, Torrey Pines Road. And this is the time when La Jolla and Torrey Pines was just exploding. There was lots of money, all kinds of ideas. People were starting companies. Phil asked me to come join Vical and set up a skunk works to carry on what I'd been doing at the Salk. So I brought my reagents, the protocols and everything, started making the RNA again. Phil set up a collaboration with John Wolfe at University of Wisconsin, who just left Ted Friedman's lab in San Diego. So that was the connection. And John had access to the use of rodents. So we began injecting the polynucleotides into rodents together, RNA and DNA, together with the cationic lipids. And lo and behold, that worked. We were all amazed and excited. It was a very heady time. However, we had a naysayer. The chemist that was making the lipids was named Raj Kumar. He now, last I heard, he ran a transmission shop in San Diego. He ended up being asked to leave the company. I don't know what happened, but it's a great shame because Raj insisted that we needed to do the negative controls of just the RNA, just the DNA, and lipids alone. And so to placate him, because I knew that wouldn't work, I'd tried that with the Xenopus and with the chick embryos, but to placate him, I sent off the samples to John Wolfe, and those were chloramphenicol acetyl transferase samples. He didn't believe that luciferase would work very well in animals at the time. John didn't. And so a CAT assay, this is the acronym for chloramphenicol acetyl transferase thin layer chromatography. So a autoradiogram was sent back from Wisconsin with the initial data. And John was very excited because it had worked yet again. I pointed out that the negative control was positive. John explained that his postdoc must have screwed up and misloaded the lane. It was rerun, and that was the genesis of naked DNA and RNA, which came out as a science paper. The prior work had come out as a PNAS paper with Inder and Phil and myself as first author. And then the science paper came out, and that was interesting because it had been just after the discovery of cold fusion. And so there was worldwide press about how this was yet another case of science gone awry. And these crazy people from San Diego were claiming that they could make vaccines and such like with RNA and DNA by just injecting the polynucleotide alone, which obviously could possibly be true. And yet it was reproduced all over the world, and that gave us DNA vaccines. So that's the genesis.
That was in 1989 or 1990?
Yeah, that was 1990, and the PNAS paper, I think, was published in 88. The patents were all filed, I'm pretty sure, in 88. And there was a large number of patents. This is another part of the story. So people ask, why did it take 20 years between when you guys made these discoveries and actually did the first reduction to practice of RNA as a vaccine and then DNA as a vaccine? The answer is that these were broadly blocking patents. They were, you can look them up, they're clearly there. The academics don't cite them, they overlook them, but they're all there. And as an industrial scientist, I had signed agreements that I couldn't work on this anymore. Everything belonged to the company. So it came out in the patents, and those patents were very aggressively defended, first by Vical and then by Merck. Merck licensed them and took credit for a lot of the discoveries. That's another story. But you can talk to Doug Richman about that. He still has some hard feelings. Doug was the influential, I'm not sure if he was a founder or a key member of the scientific advisory board, but he was very involved in the reduction to practice with influenza. But Merck tried and tried, spent a lot of money, could never make a product. And so they gave the technology back to Vical under the terms of their agreement. And Vical continued to try to develop DNA drugs. They focused only on the DNA, but aggressively kept everybody else from using that intellectual property for RNA or DNA. And so you ended up with a 20-year period and a story that's fascinating from CureVac about how they independently had come to these discoveries many years later, started developing the company. I believe that they were unaware of the intellectual property and the prior work. I'm sure they're reporting this in good faith, but as they tell the story, they were going along merrily developing RNA vaccines, and then suddenly this swarm of lawyers attacked them and blocked them from being able to do it. Well, that was Vical on the base of those patents. And the problem is that Vical had the intellectual property. They didn't have the capability or the talent to move it forward, but they were quite glad to keep anybody else from developing products. And that's why you have this dead period of 20 years. So thanks for letting me tell that story. I hope it wasn't too long, and I hope your audience will find it interesting.
Well, I personally think it's very interesting because we've heard a lot about it, but we've never managed to actually piece it together in one spot. So thank you very much for that. Now, of course, it was put on hold on top of a shelf, and then suddenly it actually comes, we're in 2020, and we all hear about RNA vaccine. In between, I would say in the last 10 years, we've heard a lot that RNA vaccines or treatments had been tried for cancer. And yes, my understanding is that there is very little ground for success. Now, is it from your point of view that they're doing it wrong, that they're researching it wrong, or is this something that you would have expected?
So this is thank you for this question. So I'm a physician scientist. I'm trained in clinical development, regulatory affairs, all that stuff. I'm not just a bench scientist, and that's how I make my money these days. And ever since 2000, I do a lot of biodefense work, and I'm a consultant. And I often get asked, well, could you help support us on this oncology project? And my general answer is, I'm sorry, I do the easy stuff. I work on therapeutic or prophylactic vaccines for infectious disease. Cancer is really complicated, and you need to go hire somebody who's a specialist in oncology. In my experience and opinion, cancer biology and cancer immunology are among the most complicated topics in the world right now, intellectually. And there are many brilliant scientists, Carl June comes to mind, many others, that are advancing the state of the art in that area. And there's also many brilliant scientists that are very well funded that have never met their objectives. And when I see that in a field, I think to myself, well, I'm not any smarter than they are. And I usually have a lot less money. And I prefer to stay away from the hard things and focus on, the phrase, the low-hanging fruit. And I think that, the likes of Novartis that has this massive amount of money, and they can choose a therapeutic target based on what they perceive the market to be, they have the latitude to explore the biology and immunology in a very great level of detail, but they're still not coming out with the solutions either. So when I see that, I say, if I'm going to be doing a little startup, and I've done a few, this is probably not a very wise business plan. So I'm not saying it wasn't a wise business plan for Moderna or CureVac or BioNTech, and I wish them the best of success, and I hope they do succeed. They haven't succeeded so far, and I don't think it's a fault of the technology. I think it's that the topic area, the therapeutic area, is very complex. And we don't understand it well enough yet, in my opinion. And I applaud the courage and fortitude of people who wish to try to solve those problems, but I prefer to stay with the easy stuff like prophylactic vaccines for infectious disease. That's hard enough. Good enough for me.
Thanks, Robert, for that. But let's move to the to this RNA vaccine. Of course, you've come out of the wood, as you say, and you've been quite vocal about the use of the technology and the side effects and so on and so forth. And since you're a consultant and you were at the origin of this, so nobody consulted you or went and sought your advice on those subjects?
No. I've had no contact from Moderna, BioNTech, or CureVac, with the exception of Katie Carrico, who I have periodic correspondence when I ask her to stop asserting that she was the original inventor. She's a vice president at BioNTech. Other than that, I have had the privilege at a few academic meetings to hear my papers cited on occasion in presentations, and I'm very grateful for that. I really am grateful for those who don't write me out of history. But yeah, through this outbreak, so this is not, I'm an outbreak specialist. I was very involved in helping bring forth the Public Health Agency Canada VSV vaccine and getting Merck involved. And we now call that the Merck Ebola vaccine. I've been involved in many vaccines, but I'm usually kind of behind the scenes and focused on my clients and meeting their needs, often Department of Defense related. So I'm not surprised nobody reached out to me. I'm not offended. It's just the way things are.
But when you're looking at the way they've actually applied and rolled out the technology, of course, you say you're very grateful to people who've plowed on with this technology and who are rolling it out big time. But at the same time, you've been pretty challenging with their application, and especially the genotoxicity of the spike protein and so on and so forth. So can you tell us?
Yeah, let me tell you why that happened. Yeah. And suddenly I find myself on the world stage with people such as yourself that normally would not even know who I was, asking for an interview with me. And you're not the only one. It's almost continuous, these requests for interviews and comments. It was another one of these cascades of events. I am very rigorously trained in bioethics. And I guess if tracing it back in my mind, I was on a phone call with a Canadian physician for a few hours who told me of his experience, and I was already aware of some of the adverse event noise. And I have very good contacts within the FDA. And I meet every other week or so with some senior people at the FDA, and we just kind of talk back and forth about repurposed drugs, about the vaccines, about the pathogen, et cetera. I'm very knowledgeable about what goes on in the government. It's important for my business to be so. But I had this call with the Canadian physician who told me about what he's experiencing, in that he's reporting adverse events. They're immediately being dismissed. Other Canadian physicians are being terminated if they speak out about their concerns. And I was aware of some of the censorship, even in France, particularly for those who wish to explore, physicians who wish to explore alternative treatment strategies. So that was my background. We had this conversation. He told me about all these adverse events that his patients are experiencing and what he is experiencing as he's reporting them into the French system. Told me about the situation in France in Canada, with the government doing things like using ice cream cones to try to get children to come and be vaccinated even without their parents' approval. And I listened to these horrible stories, what I thought were horrible from a bioethics standpoint. And I told the gentleman, my colleague, physician colleague, I just didn't know what I could do to help him. And then I went to sleep that night, woke up in the morning, and I said, I know what I could do. I could build a paper that described the bioethical considerations that need to be applied in the context of COVID vaccination when these are experimental vaccines. So I wrote that paper based on the Nuremberg Code and all the subsequent knowledge and concurrence that we have in the West about bioethics for clinical research, because remember, these days, that's what I do mostly, is clinical research. And I posted that on a American media site called Trial Site News, very industry insider focused on pharmaceutical industry and people that do clinical research.
I read the paper, yeah.
And it got a lot of attention. And I started getting people writing to me from all over the world saying, thank you so much for doing this. Then I was asked to participate in a podcast in the United States from a gentleman named Brett Weinstein, who operates in this interesting inner space. They call it the dark web. It sounds very spooky, but what it means is it's people who aren't politically affiliated. They're free thinkers. They are exploring ideas, intellectuals. And I sat in this 3-hour podcast. 3 old men, who would have thought? 3 old men sitting around a table talking to each other about things like a 40-year-old anti-parasitic drug called ivermectin, and concerns about database structures and safety signals. And it went viral, and it had almost 800,000 hits within a week before YouTube censored it and took it down. And that set in motion a cascade of events that catapulted me on the world stage. And I didn't seek it, it happened. But with that came the combination of these events and my legitimacy with my CV and my history with RNA vaccines, and kind of the genesis of this whole idea, tracing back to the Salk. Really the adenoviral vector and the mRNA vaccines have this common source. They're both gene therapy applied to vaccination, which was the idea, that the brainstorm that I had when I was 28, back there in the gene therapy lab. So I do have detailed understanding of the technologies. And by the way, I just want to shout out, you mentioned suddenly we have these things working. I did many, many years of research as a discovery researcher in the liposome field and cationic liposomes. One of the people whose work I've always respected is Dr. Peter Cullis of the University of British Columbia. And if there is an enabling technology here that transformed this field, in my opinion, it's the work that finally matured with Peter and his team with ionizable cationic lipids. And then he's licensed that out to the company Arcturus. That technology, those lipids are what underpins the CureVac product and the BioNTech product. And the Moderna product is basically reverse engineering the same thing. So I think we need to recognize the importance of Peter Cullis, who's another person that doesn't seek the spotlight. He just quietly does his work for 40 years and then the world benefits. So I had this platform suddenly, and I was being contacted by physicians all over the world, and scientists and patients. The patients just call me continuously about their personal syndromes. And mostly they need to be listened to and acknowledged that they're not crazy, because of all the censorship and all the messaging that these vaccines are completely safe. They feel like there must be something wrong with them. They must be crazy because the rest of the world is all saying these are perfectly safe. But I'm a seasoned vaccinologist and pharmaceutical developer, and I know that there are no perfectly safe vaccines and there are no perfectly safe drugs. And what I find is that with all of this messaging, and I'm sure it's all for the best intentions, many governments, certainly my government, in my opinion, is not being open and transparent about the adverse events. Now, do I think that I want to say, am I an anti-vaxxer? Am I anti-vaccines? I've spent my whole career developing vaccines and testing them. And, okay, I don't know how you can say that. However, I feel very strongly that I'm pro bioethics. I'm pro transparency. I'm pro honesty. And I am absolutely insistent on evidence-based medicine. And what I think I've seen, not the first time, remember I've been through a lot of these outbreaks, is the substitution of expert opinion, let's call it, for evidence-based medicine. And that always happens at the beginning of an outbreak. It's perfectly understandable. We never have enough information about the pathogen and about what's going on. You're surrounded by the fog of war. I could give you story after story. I have, for people that in public health, I really don't want to second guess them. I've been there. It is a rough thing to do. However, that was then. And now we're a long ways past that. We know a lot. We know a lot about these vaccines that are still there as emergency use authorization, non-licensed medical products. And I know that we have these fundamental rules of bioethics. These are experimental products, therefore they fall under the guidance for experimentation on human subjects. I don't see how you can come to any other conclusion. And the rules for experimentation on human subjects that I think all of Europe and the United States and North America can agree on. I mean, there's some countries that are under authoritarian directors, and I can't speak to them. But for those of us that share this common belief in ethics and medical research, we can agree certainly on three core principles. One is there has to be full and complete transparency and disclosure of risk. This is fundamental, and we all know it. Every time you open a package insert when you buy a box of medicine, you see a huge list of risks, and those have to be disclosed down to a very granular level. Those risks are not being disclosed to patients right now. That bothers me. Second thing, those risks have to be described in language that everybody can understand. This is written into the US law. In federal law, we call it the common rule. I have to live by these rules as a clinical developer, as a principal investigator. Okay, so the risks have to be disclosed in a way that they can be readily understood. And the third key point is that there must be free consent that can to participate in receipt of the investigational product, and that consent cannot be coerced. And I don't think there's any debate there. We all agree on those fundamental principles. Now, if you say, well, we're in the middle of a crisis and we have to waive fundamental human rights and fundamental foundations of medical ethics, I think we've crossed a line. And I really think, I really think that we need to take a step back, take a deep breath and say, what are we doing here? We don't torture people. We don't use internment camps. When we have made those kinds of decisions during times of war, we've ended up regretting it. People talk about outbreaks and pandemics as being like war, and in many ways they are. But that doesn't mean we should jettison our ethics, in my opinion. I believe that people have the right to choose what to do with their own body, at least in the West, particularly relating to experimental drugs. And so as I've been thrown onto the world stage and my colleagues have sought for me to speak for them because they are getting censored and they can't speak often. Another physician has been fired in Canada. One is being run out and they're trying to take his grant money away. Dr. Bridle, that you may have heard of.
Yep. I actually wrote a paper on him and I was fact checked. And basically they told that he was, how can I say, mad, crazy, or whatever you want to actually use as terms to discard his opinion. And so it's interesting to get your views on his papers and his point of view, especially because he spoke about the fact that the spike protein was cytotoxic, and that the manufacturers probably didn't do the job of disclosing some of those documents of
so thank you for that. And I was on a podcast with Dr. Bridle. I found him to be exemplar of the best of Canadian science. He's a modest person. He is deeply committed to academia and integrity and ethics, as I am. He felt compelled to share his interpretation of a common technical document that was obtained from the government of Japan, the regulatory authorities, that was the CTD for the Pfizer vaccine. Most of it's in Japanese. On my side, I was asked by Trial Site News to review this document that Dr. Bridle and his colleagues had obtained. I think I got it within about a day, as well as the EU EMA response letter. Presumably, I infer, to the same similar common technical document, probably in French or German or English, that was submitted to the EMA. So I received these two documents to evaluate. I independently made my own assessment. Remember, I'm fundamentally a gene therapist and the guy that came up with this stuff in the first place. So yes, I saw it from a perspective that might be different from the usual vaccinologist. And I think that's a core problem here with the regulatory agencies, is they are not looking at these agents as both vaccines and gene therapy agents. So that's just my editorial. They are both, and you have to be aware of that. These are not traditional vaccines. In any case, I reviewed the Pfizer document, the EMA letter, and I was a bit alarmed by what I found and surprised. Pfizer is a high quality organization. I expected a more professional piece of work, but what I found was the biodistribution studies were non-GLP. Your audience, I'm assuming based on your expertise, will understand these words. Stop me if I'm using too much jargon.
The GLP is, sorry, you say non-GLP?
Yeah, it was non-GLP for the biodistribution studies. And as someone who helped pioneer luciferase, they did a very odd thing. To look at the distribution of expression of the transgene, they used a surrogate, not spike open reading frame. Normally, for a GLP tox biodistribution study, you would administer essentially the final drug product, which would be the RNA encoding the spike, wrapped in the necessary lipids, etc. Instead, they elected to use a luciferase RNA. This is the open reading frame that makes the firefly tail glow. It produces photons. They're very easily detected. Normally with the luciferase assay, if you're going to look for distribution of gene expression, you dissect the animal, homogenize the various tissues, and then count precisely the amount of luminosity you get in the form of photons and convert that to picograms of protein, or femtograms. In this case, surprising to me, Pfizer used the least sensitive method, which is whole animal imaging, to detect the distribution of the gene expression. Now, a cynic might say that they were gaming the regulators by using the whole animal assay, but for whatever reason, that is the least sensitive way to detect luciferase. You're requiring that the photons travel through the tissue, through the bone and muscle, through the skin, through the hair, and be detected by a luminometer or a photomultiplier tube, essentially. Okay, and you can image the whole animal, and not surprisingly, all the signal is detected right around the site of injection. Okay, so based on that, a regulator might concur that the complexes stay put where they're administered in this non-GLP rodent model. They also did two other studies. with biodistribution, non-GLP, where they looked at the distribution of the lipid component. So people like the metaphor of a letter in an envelope as a way to deliver RNA. RNA being the letter, the envelope being the lipids. So in this Pfizer CTD package showed a study wherein the lipid component, its distribution in the body of this small number of rodents, again, non-GLP study, was measured, the distribution of the lipid, and sorted by organ. So they analyzed the organs, just as I said they should have analyzed for luciferase. They did this for the lipid delivery. And they also tritiated, so they radioactively tagged RNA, put that in the formulation, and looked where the RNA went. One of the things that was a bit surprising, the RNA did distribute in part, and the majority of the RNA and the majority of the lipids stayed put around the site of injection, but there was a significant amount of both that was distributed through the body of the animal. Not surprising, a lot of this ended up in liver and spleen, places you would expect, for reasons that are self-evident to anybody in the industry. But we also found lipids in bone marrow. And most surprisingly, we found about 12% of the lipids, or the Pfizer data indicate, 12% of the lipids end up in ovarian tissue. That is not the case with testicular tissue. So this was not only a striking apparent accumulation of a novel pharmaceutical reagent in a female reproductive tissue, but it wasn't being concentrated in the testis. So that got various people alarmed, including Dr. Bridle. Now, I'm not sure, there's been a lot of confusion about this, and perhaps I'm able to see it more clearly because I understand the technology and the assays, etc. But I think it's really important. I'm hearing people say, well, this shows there's gene expression in the ovaries from the transgene. No, it does not show that. It shows that there is a novel lipid, which is the packaging material, that appears to accumulate in ovaries and not in testis, and also is present in bone marrow and some of the other organs that you might not expect it to be. It's not the majority of the lipid, but it's not a trivial amount. However, it's in a non-GLP assay with rodents. Okay, so if we're going to criticize them for not doing a fully GLP tox study for biodistribution, we can't then turn around and criticize them for the results performed in a non-GLP study. So, but those were the findings. And then I took this and shared it with a more senior regulatory official who wishes to remain anonymous, but is president of a large European society. And asked him, I said, could you look at this? Because I'm a little concerned here and I've got to say something. I've been asked to review and opine and about these data. And would you just cross-check for me? So he did, and he looked at the EMA document, and he noticed that, in fact, the full spectrum of reproductive toxicology studies had not been performed, nor had the genotoxicity studies. Okay, so now, normally you don't have to do full reproductive toxicology and genotoxicity with a vaccine. I spoke about this with my friends at the FDA last night. I called them together and I said, we need a special session. And I need to talk to you about what I've been saying and what we've been seeing. And I'd want to get your opinion on all this. And also your opinion on what's going on with the databases, the safety databases and all that. So we talked about all this. And two of the three of them concurred. One was alarmed that I was speaking to conservative media in the United States that was going to weaponize this against the administration, but none of them criticized the observations. So I think in sum, we need to not hyperventilate. These are non-GLP studies, and I found out subsequently in speaking, so Peter Marks, the director of CBER, granted me an audience, and we spoke for half an hour over Zoom together with his director of external relations for CBER. And to paraphrase what he said, this is not a direct quote, was we received another CTD package from Pfizer about 2 weeks ago. And at the time when we had the call, and he said, give us the opportunity to review that package. I don't see anything there that has me alarmed, but I haven't read it myself. Okay. And I said, you know, out of professional courtesy, he's got a great reputation. He's a smart man. He's known for his integrity. And I think we need to kind of back off and let him do his job. And I think it's the appropriate position for me and for all of us to allow the regulators to review these new documents that they've received from Pfizer. And I hope that Pfizer has done reproductive tox and genotoxicity and the GLP biodistribution studies and all that. I think we need to provide the benefit of letting them do their job. That's my opinion. Now, Byram is, Dr. Bridle is, this is not his sweet spot, gene therapy and these new technologies. He reviewed the same data. He came to the same conclusions. Maybe there was a little less clarity in some of those conclusions in his mind. That's not surprising. He's not an expert in this little niche. He felt it necessary to alert the world about what he saw. And I'm positive he did it out of his own personal integrity and a sense of obligation. For me, I tempered my communication quite a bit, knowing how explosive this could be. And I've continued to do so. And again, I want to emphasize to your audience, these vaccines are saving lives. I'm not saying that they are unsafe. I'm saying that we don't really know how safe they are. And the authorities often are saying these words, risk-benefit ratio. But for those of us who do this for a living, that's a very precise term. Risk-benefit ratio is something that is calculated. It can be quantitated. It's typically calculated for each of the special populations like adolescents, children, infants, pregnancy, elderly, etc. So just these blanket statements that the risk-benefit ratio favors vaccinating adolescents, that if someone wants to say this, again, let's go to evidence-based medicine. We don't need opinions. Show me the data. Show me the calculation. And I'm glad to say, oh, this risk-benefit ratio, after you do the calculations, it makes sense to vaccinate 20-year-olds or whatever the question is. But when somebody asks me, as people are, should we vaccinate children? Well, I don't see clear evidence that that makes sense. And I am aware of the safety signal. Now we have the official statement, but I've known about it for a couple of months now, about the different cardiotoxicities, pericarditis, etc., in adolescents. I'm concerned that if your audience understands biostatistics, they'll understand the term confounding variables and masking. And if we're looking for cardiovascular events in a general population, it's going to be very easy to find those events associated with a treatment in adolescents because they have almost no cardiac disease. As you get older, you have masking. It becomes harder and harder to get that signal. So I'm not confident that we have ruled out that those effects don't extend through other age groups. I'm just going to say that. I'm aware that there are other safety signals that are not yet disclosed to the public. Some of those were disclosed in a recent Wall Street Journal editorial by two well-respected academic physicians. One of those is thrombocytopenia. There's others. One of the ones that hasn't been disclosed in press that some within the FDA are aware exist is reactivation of latent viruses. Now, how serious are these? My point is we don't know. And right now we can't know because the databases at the FDA will not support a rigorous analysis because they are very small, self-reported, and not really well characterized. The FDA and the CDC had relied on the Israeli database, which is more comprehensive, but the Israelis didn't detect the cardiac signals. So then I'm a little more wary about that. We had the thrombotic events associated with the adenovirus vectored vaccines, and Norway is particularly sensitized to those right now. And the Norwegians, as I suspect we'll see with the other Scandinavian countries, are doing a good job, I think, with their data capture and analysis. And I suspect that we will know more about the severity and distribution of adverse events as Norway and the Scandinavian countries deploy vaccine. But I'm aware, and the literature shows, that both RNA and adenoviral vectored vaccines expressing spike are associated with thrombosis. And here's the thing. I mean, we're all scientists here. This is a physician-scientist discussion we're having. The adenoviral vectors from Crucell and others have been used to develop many, many different vaccines. They've been in many human patients for over a decade. We have that safety database. I don't recall hearing that thrombotic events were a major problem with adenoviral vectored vaccines. We do know that thrombosis is a significant problem with COVID, with the disease subsequent to infection by SARS-CoV-2. We see thrombosis in the adenoviral vectored vaccines, which probably express the protein for a longer period of time and at higher levels than the RNA does. We don't actually know, by the way. So that's not a proven, but that clinical finding is there. And now there are reports coming out from all over the world, and recently one from Singapore published in the American Journal of Hematology, of central cerebral thrombosis at an unusual rate in Singapore after receipt of an RNA vaccine. So I've been fact-checked saying spike is not toxic. Well, my statement was that the native spike protein is toxic. It does open the blood-brain barrier. It does cause cytotoxicity.
By native, you're talking about the spike protein of the virus.
Precisely, okay? And these data, this cytotoxicity and blood-brain barrier effects tested in organoids and in mice, and demonstrated. That data didn't come out until much later after the vaccines had been designed. So there are those who assert that the NIH Vaccine Research Center and the other centers that developed these vaccines engineered them so that the spike would be safe. It's hard for me to understand why they would have done that when they weren't aware that they were not safe, that the spike itself was toxic. So unless they have a time machine in Germany and in Boston and in Bethesda, Maryland, I don't think that makes sense. I think that, for instance, they engineered the receptor binding domain to be locked into the open conformation, not for safety, but for immunogenicity, to elicit better antibody responses against that domain. I think that's why they did it. Now, on top of this, there comes unexpectedly the nurses' study from Harvard and Brigham Women's that shows after vaccination with Moderna, which has the highest dose of RNA, far and above, 100 mics, instead of the Pfizer dose or even the CureVac dose, which obviously only gave 40% protection. using a very sensitive method, it was possible to detect free spike subunits circulating in the blood of these nurses after vaccination for almost up to a month in some of them. And the levels aren't trivial. When I read the levels and I did the calculations, I thought, hallelujah, Peter Cullis has done an amazing thing. It is amazing these levels of protein expression that you're getting from RNA after a non-viral gene delivery. However, with that success, like everything, comes a warning. Spike is biologically active, and those who say that the vaccine expressed spike does not have the biologic activity of the native spike, I challenge. It's like the French system. My metaphor is the French penal system that assumes you're guilty until you're proven innocent. That same rule applies with pharmacology and toxicology. The assumption has to be that the new product is not safe until it's proven safe. And so those that say, well, they must have done something to make it safe or different in the vaccines, my response to that is, I hope you're right, but I haven't seen that publication. So if somebody knows of the paper, peer-reviewed, published, that's our standard, or the CTD that shows that Pfizer, Moderna, CureVac, or whomever, or J&J, have done the toxicology and demonstrated that the spike expressed from their vectors does not have these same biologic effects of opening blood-brain barrier and causing cytotoxicity, please share it with the world, and I will be quiet. But in the meantime, I do think that Dr. Bridle is correct. There is a suggestion of risk. It's non-trivial. It's consistent with some of the signs and symptoms that we see in this post-genetic vaccination syndrome that some people are reporting. How severe is it? It seems to vary. How frequent is it? I don't know, because we don't have good databases. Is it affecting the risk-benefit ratio? I can't say, because the data aren't available to evaluate. And that's my objection here, is that there's a need for transparency and openness and allowance of scientific discourse. The tension that makes for good science requires that we talk to each other, and that we admit our potential problems and we address them. Well, there's another one that bothers me a little bit, if you'll allow me, and that are the assumptions about what it's going to take to reach herd immunity. Okay, and this bothers me a bit too, because I'm trained in epidemiology and I understand about herd immunity and vaccines. Herd immunity is a point in which we're no longer having spread of virus between individuals in a population, because we have done a series of interventions, masks, whatever. Okay, if we're going to say that 70% of the population has to take one of these vaccines to reach herd immunity, in order to do that calculation, we would have to have data on the effect of the vaccine on virus transmission. But those are not the endpoints that we've been sampling. We've been sampling disease endpoints. Do they keep you out of the hospital? Do they keep you from dying? Those are really important endpoints. I don't deny that. But they're not the endpoints that will allow us to calculate herd immunity. So let's also be a little honest about that, please, and fact-based. If we are going to use the vaccines and try to implement requirements for a population that they have to take so much, certain amount of people have to take vaccine, well, let's make that science-based. But I know that we can't right now, because we don't have the data, because the clinical designs for the studies were not for transmissibility studies. So let's do those studies. And then we can have a real discussion about how much vaccine is needed versus repurposed drugs or other things. But my point is, let's get to using data.
But I mean, of course, you mentioned a lot bioethics. You've given us a sort of with great details what you think are some of the issues. But we're still seeing, I would say, government agencies health authorities around the globe who are promoting those vaccines, who are pushing for more vaccination. We've seen the ice cream issue in Canada. We've seen $1 million offer, a raffle to actually get vaccinated. Clearly, these are marketing devices, and there seemed to be a contradiction between the incentives that they're using versus the risk that you seem to be portraying. So it's only time to take a pause and basically try and assess what we're doing, complete the genotoxicity analysis that we're not sort of remitted to the FDAs or to the EMEA, so that at least we learn from what has been achieved so far.
So I think what you're saying is it time to put these vaccines on hold while we ask for those more complete data packages?
It's a combination of that. You
that.
talked about early treatment as well. I mean, surely when you have
Yes.
the response of an to a pandemic, you have a certain number of arms, should I say, or measures you can use, like social distancing, early treatments. I would say treatment, and then you have vaccine. And we seem to have focused all our effort onto one. And I spoke to a lot of doctors who are in favor of ivermectin or azithromycin or zinc or early treatments, and
many things
seem to have been censored. You know, they seem to be to have double standards applied. I mean, to them. For early treatments, they want to have the highest level of standards, and
this is I agree. This is very, very troubling what's happened here. I can speculate as to what's going on, and I prefer not to. There is a international news consortium that is very transparent in a press announcement from the BBC. It includes AFP, by the way. So there is a truth in information consortium that includes Google and Yahoo and all these others that is, in this BBC press announcement, they're actively suppressing what they perceive to be disinformation. Now, forgive me, you work with the press, but my experience with most reporters is they would not tolerate this level of discussion that you and I just had, let alone report it. Often in the States, at least, what I'm seeing is that, for instance, in this RNA vaccine story, the only people that are press people that are contacting me and doing detailed analysis, looking at the documents, looking at the patents, are European journalists. They're not doing this in the States. But there seems to have been something happen where the press has gone to sleep. And I don't understand that. But I do find it troubling, and this is why I am receiving all this information from all over the world from physicians and scientists that are also have anguish and concern about these things. In terms of, is it time to put it on pause? We also still have the risk of the Delta variant and hospitals filling up and people dying. So my position, to be perfectly clear, is that these vaccines on balance, particularly for older adults, are saving lives. Deprive them of the possibility, if they haven't already received vaccine, deprive them of that possibility. I'm not comfortable with that, but ethically. However, I am comfortable saying, let's fully disclose to people what, I just got a internet unstable, sorry. Let's fully disclose to adults and rely on them, rely on the people of France, for example, as adults, to make their own decisions about their own bodies. And if they choose to use vaccine, that's their choice. And we need to make sure that they have all the information necessary to make an informed choice. And if they choose not to use masks or social distancing or quarantine themselves in their home or whatever it is they choose to do, I believe that it's a fundamental shared principle that France championed, right? Liberty, equality, the right of individuals to make their own decisions and adults. And I believe that in the interim, while we sort these things out, we owe it to our fellow citizens to treat them with respect and give them information. Now, it comes to me, people ask, well, what do we do now? It's my opinion that certainly for Moderna, the dose is too high. It's my opinion that, and it's shared by many that I talk to, that there is data suggesting that these RNA vaccines may also be very effective when administered as a single dose. And the bulk of the adverse events and the frequency and severity are much higher with a second dose. So that would be something that could be done. I think that we're going to see the rollout of second generation products, both RNA based and traditional. And I believe that the more traditional vaccines may or may not be as effective. The Novavax vaccine is encouraging. It's not fully a traditional vaccine. It's made in insect cells and has a novel adjuvant. But the novel adjuvant is derived from tree bark. It doesn't have aluminum in it. So that's something that many people might like. And I think that as that becomes available, then for those that have hesitancy about the genetic vaccines, they might want to think about traditional vaccines. And I think we need to make those available. So I don't think it has to be catastrophic that we just suddenly turn off the pipe. I think that we do owe it to the people of France and Europe and the United States and Canada to be open about what signals we're seeing about safety so they can make their own decisions. For instance, pregnant women. It's been noticed that the CDC put out an interesting piece in MMWR a couple weeks ago in which they said, based on our vSafe database, we think it's safe for women to use these vaccines during pregnancy. But in fact, the database is so limited that we really can't be sure and we might change our mind. It was basically the summary paragraph at the end of that. Then shortly thereafter, NIH launches a prospective clinical trial of safety of RNA vaccines in pregnancy. That does not inspire hope and confidence in many people. And I think we owe it to mothers and fathers and children and future children to tell them what the data are and trust them as adults. And that's the other part that kind of comes with this censorship, is there's a sense of incipient authoritarianism that makes many of us uncomfortable. Have I answered your questions? We've talked a long time.
Yes. Maybe I do have one last question, because you spoke a lot about vaccines. We did not really address the double standard with respect to early treatments. And of course, since you say that you like to focus on simpler things, it sounds as if sort of early treatments, reposition drugs, and so on and so forth appear to be reasonably simple. Maybe it's not simple to man in the street. What are you?
Remember, remember I said I'm somebody who doesn't seek the limelight, and I try not to self-promote. Probably that's a fault. I received a phone call from an American in the government who was in Wuhan in December of 2019. He called me in the first week of January. Let's not go there, okay? We'll just park that. There's a lot going on about when did this all start and how did it start. But in any case, I got this call in the first week of January, and he said, Robert, you got to get going. And I was leading a team on behalf of the government that was screening using high throughput systems, compounds as countermeasures for chemical warfare agents. And we were using the latest computational technology and other things. And I got my team to volunteer their time to start doing similar screening and docking focused on one of the key proteins of the SARS-CoV-2 immediately after the first sequence was published. I think it was January 10th or January 11th. So we started that. We did a lot of modeling. We identified some initial compounds, screening all licensed compounds, this kind of thing, for their activity against the papain-like protease and the known binding sites for that. And we identified some compounds on the first screen. And then I was at a drug discovery conference in Cambridge at the end of February, housed right across from Biogen corporate headquarters. And I came home sick as a dog. with COVID. So I was in the first wave of East Coast infections personally. And I started taking some of those drugs that we had identified by computational screening. And I got immediate symptomatic relief with one of them called famotidine. You may know as Pepcid, the H2 blocker. And that kind of set a whole bunch of things in motion. But that group eventually became funded by the US Department of Defense. And we've carried on. I have a close collaborator who's a physician in Wisconsin named Tamara. And he took I worked with a large group working out what the mechanism of action of famotidine could possibly be. And it was nothing to do with our docking. It was a classic H2 blocker mediated pathway, which led to the mast cell hypothesis and all of these things that now at the time was heresy, but is now accepted wisdom. And very hard to publish, took me almost a year. Then Kevin found a Chinese paper talking about celecoxib in a randomized clinical trial and the biology of celecoxib. This is the COX-2 inhibitor, selective COX-2. And he combined them, and suddenly there was amazing clinical responses. So we have papers on famotidine plus celecoxib pending review right now. That's another story involving frontiers in pharmacology we don't need to talk about. And I became aware of the data on ivermectin as it was emerging from Andrew Hill and Pierre Corey and other advocates, and suggested that we start testing the addition of ivermectin on top of famotidine and celecoxib. And in fact, Kevin Tamara at Beloit generated some very nice data that suggests that added ivermectin really further enhances the clinical response, particularly in the recovery of the leukocyte fraction and particularly lymphocytes. The FDA was not sanguine on our IND that included arms with all three agents. And they insisted that we would have to demonstrate the mechanism of action of ivermectin as a antiviral for SARS-CoV-2 using cell culture experiments. And of course, that would have taken another 6 months. So the US Army decided to just drop the ivermectin arm in the face of FDA objections. We still don't have the IND approved. We've been through, I think, 4 rounds, and the team finds the FDA intransigence exceptional in their, unprecedented. Every time we answer their questions and solve the problem they gave us the last time, they come up with new ones. I've never experienced this in dealing with the FDA when you have an IND, and usually they say, here's our objections, fix this. You fix them, and they say, good to go. Go ahead and start your clinical trial. But we've been through 4 rounds of that. So I don't know how to interpret that, but it's happening. But in terms of repurposed drugs, what I'm telling you this story is that I've been very involved in repurposed drugs from the start as a vaccinologist, because I made a threat assessment in which I concluded that to do vaccine development responsibly was going to take too long. And our best option in short term was repurposed drugs. So it has been, if you'll allow me to quote the Grateful Dead, a long, strange trip. And it's not over yet. I may be able to get some trials started in India that might enroll quicker, and we might get to the endpoint sooner. The ivermectin data is, I remain intrigued. I'm surrounded by people who think it is the second coming of Jesus, and I don't. I think that the data, the meta-analysis data with all of its warts and flaws suggests that it is reasonably likely to be demonstrated to have both prophylactic and therapeutic activity. But does that mean that it reduces the incidence rate as a prophylactic of hospitalization by 1 in 5 or 1 in 3 or 1 in 2? I don't know. Those data aren't here yet. It is not a silver bullet that's going to be the same as a 90% effective vaccine, in my opinion. I could be wrong. I hope I am. I hope it is 100% effective. I doubt it, And like the rest of the field of folks that were, more grounded in traditional pharmacology, which I also know a fair amount about, the imbalance between the measured IC50 and the Cmax at safe doses doesn't make sense that it would be effective in at these dosages as an antiviral. And so how is it acting? I personally think that the activity we're seeing may have a strong component of anti-inflammatory activity. And I think that COVID is a disease not of virus infection, but of virus-triggered host immune response dysregulation. And I think that the data to date with the antivirals being so disappointing suggests that we really should focus on mitigating the disease as a hyperinflammatory disease. The problem with that, and the problem with the logic that some people put out about ivermectin being our solution, that we can eradicate COVID with ivermectin. You hear these words. Those people that say that haven't typically thought through that we have no data demonstrating that ivermectin is reducing viral shedding or transmissibility, just the same as the vaccine problem. And their data, the data that Tess Lawrie has published and Andy Hill is accumulating, is also largely based on disease endpoints, not viral endpoints. And so if we agree that herd immunity is going to be a problem of blocking viral infection transmission, but the disease endpoints are problems having to do with mitigating the hyperinflammatory response after you've been infected, then those endpoints don't predict what we need to control the virus in terms of herd immunity. And I think we have to be honest about that too, with ourselves. So that's my kind of quick version of quick travel through how I see the repurposed drug space. I see a lot of hope in repurposed drugs in mitigating the hyperinflammatory disease that kills people, because we have lots of great drugs for inflammation, thank heavens. But the antivirals have generally been disappointing. Maybe the new EDD 2801, however Merck has renamed it, that crosses the blood-brain barrier, maybe that's going to work. There are naysayers that suggest that it may be have reproductive toxicology problems. The data will show, but history has shown, Tamiflu being the poster child, that antivirals for respiratory pathogens, viral pathogens, generally don't fulfill their promise. Once a virus gets its hooks into you, it's hard to clear it. So you asked about my opinions about repurposed drugs, and now I've lived it, I've walked it. From the very beginning, I've been right at the forefront on this, and I'm hopeful. I think that we do have agents that show considerable promise for the disease. I don't have any data on whether we have agents, both new and repurposed, that are really going to allow us to come up with ring containment or other strategies to result in eradication within a region.
Robert, thank you very much for all of your time on this. It's been really great, very thorough, because you're taking the time to provide us with the flow of your thinking and the flow of your experience. I probably have one last question, because I do ask this question to most of the doctors that I actually interview. Everybody talks about conflict of interests or links of interest. Do you have any specific conflict? Because you've been involved in the vaccine industry, you've worked with also repurposed drugs. I suppose you don't, but I mean,
well, let me say it. And I endorse this. It's a really important question. And the journals have become more rigorous And I endorse that. I find it burdensome because I have so many potential conflicts of interest that I have to disclose, but I do that. You can read my papers in Frontiers or the ones that are online pending in the preprint servers, and you'll get a nice summary. So I serve. One of the things with these repurposed drugs that we're working with, there was a conscious decision by the team to not patent anything we did. We intentionally put everything that we did into the public domain. I believe in the vision of Médecins Sans Frontières, and I work with MSF. I believe that in a time like this is not the time for profiteering. So all of that stuff is public domain. No patents have been filed. I do work for various companies that are trying to develop products that relate to COVID. None of those are matured. We haven't talked about any of those today. They're biologics or other antibody or other types of products. I have one client that I'm not supposed to disclose. It is the largest conglomerate in India. And that very large conglomerate, headed up by a very wealthy man, has a particular focus in their new vaccines division, which I serve as a consultant to trying to develop a COVID vaccine that can address the needs of India's rural poor. So that means a vaccine that has to have a price point at below $1 because we're talking about, pretty close to 1 billion people. That's a lot of money. And so I do support that team. They're developing a subunit-based fermentation grounded vaccine that has a more traditional adjuvant. So very much an old school vaccine. It's about to enter into initial clinical trials. I've helped them with the protocol and reviewing the protocol. So that could be interpreted as a conflict of interest. I hold no stock or have no financial interests in anything to do with Moderna, CureVac, BioNTech, Pfizer, Merck, any other pharmaceutical company. I hold no stock. And none of those that I just named are clients, nor is Novavax. Once upon a time, the two leaders of Novavax were my clients at a prior company, but that is over a decade ago, and I know them well. I know the good and bad of Novavax, but I have no ties with them. So there's my COI. I hope that's comprehensive enough.
Well, Professor Malone, thank you very much for all of your time today. It's been an honor and a pleasure to, in fact, learn more about the RNA sort of history. And I just want to say thank you on behalf of all the viewers at France Soir, and wish you to have a very good day then.
Thank you. And thank you for the opportunity to speak to your listeners at France Soir. And I hope I am scheduled to attend a meeting in Nice in the early winter. And I hope very much I'm going to be able to do that because I look forward to it.